So I would like to welcome all of you today. It's a big privilege and for me to welcome all of you. This is a combined organization with the Semmelweis University Center for Translational Medicine, the Academia Europaea Budapest Knowledge Hub, and the National Academy of Scientist Education. So I would like to warmly welcome our guests who are here today and give us the pleasure to welcome there. So I would like to introduce first the program, what we are doing, and later on we will have the seminar lectures. So actually this PhD program you are there is a clinical translational medicine program, but I think it's very important first to mention that to translate anything for clinic, you need to discover something first. So science, you need to start with science, but this scientific activity needs to bring back definitely for society's benefits. So how is science is going? So first of all, as I mentioned, you need to discover something. And if you discovered many things, of course, sometimes you don't even know what benefits this discovery will bring. But if a healthcare challenge comes, including cancer, COVID-19, dementia, anything, you will have a tool to use to treat patients. But what you need to feed science, you need young, talented students and creative ideas. So, but why we established this National Academy for Scientist Education is very easy. Here are statistics data among secondary school students and the interest in sciences in the recent decades. And is definitely decreasing even in Asia, Europe, Middle East, and all countries. So you need to do something to make the young people to get interest. However, when you have a huge pool of scientific results, you need people who are doing the healthcare service, including doctors, dietitians, psychologists, pharmacists, dentists, who will actually bring the scientific knowledge back for patients. Why do we need to take attention of it? Is it a problem? I just show you statistical data for you from Eurostat. If we stop science today, and there would be prohibited to do any science in medicine, but the human being would use all the scientific knowledge which were generated in the past, under 75 annually 1.2 million deaths could have been avoided or delayed because, of course, deaths cannot be fully avoided. And in those countries, even where the scientific activities lower, the loss is higher. So in Central and Eastern Europe, 4 of 5 deaths under 75 could be either prevented or treated. So therefore, we need specific programs for healthcare professionals who, after the discoveries, understand how to translate the scientific knowledge back for patients' benefits. And of course, we can't solve our problems with the same thinking we used when we created them. So this is why we need new educational models. So therefore, we had 2 very distinct specific goals. One of them, we so-call it from curiosity to career. This is for young people to learn scientific activities. And this is what the National Academy of Scientist Educations are doing. Yesterday, we visited secondary schools and looked at how the program runs. Currently, there are over 3,000 high school students are involved in the program. We have a 20-year plan. We are about in the half line, but we are definitely will not stop. But the second program, and this is where we are currently, this is from science to society, and this helps to bring the knowledge back for patients' benefits. This is what we established first at the University of Page and brought here. Now we have within 3 years, 352 PhD students from 22 countries. And I was really very pleased that the whole methodologies for the whole education system one of the prominent journals, the Nature Medicine, found it for publication, but we discussed with Marty also these kind of journals. We had to change the title many times, you know, to have suitable for the internal editors, because we also put the systems education, that's a new model, and they didn't like the word, actually, the new model. So this is the program, the Science to Society. This is the number of students currently involved in the programs, and after Semmelweis University, the number of students really elevated, and I'm so happy that currently there are students, and around here as well, if you look around, this is completely international. We have here over 24 countries are there. But just briefly, what is this program about? Because in the discovery, you have to wait plenty of years to discover something. But when you do a clinical science, this can be translated even earlier than somebody received, for example, a PhD. So here is one of the issues. In pancreatitis, some places over 70–80% the usage of antibiotics. So a PhD students came to Page and learned what the different biomarkers mean, the CRP, leukocyte level, procalcitonin. I realized and learned and published that this is not good how we use. And when she returned to the Székesfehérvár Hospital, she immediately started using the knowledge, what was generated from clinical science. And within a single year, she could decrease with 31% the usage of antibiotics and the length of hospitalization with the patient, and she defended her PhD 1 year later. Another example from the Heinz-Pahl Children's Hospitals, where in CF, cystic fibrosis, we know that after a certain age, after year 10, the kids can get diabetes as well. And plenty of times this is recognized when they have visual impairment in the school. So what they did, they started screening in their PhD program at the age of 4. And within 1 year, they could identify 30% of the kids with prediabetes or impaired glucose tolerance, and they immediately could make intervention before the complication system started. And after Andrea Parnitsky defended her PhD. The third example, an otorhinopharyngologist from the hospital came to me that they operate cholesteatoma plenty of times, but this is also recurring. So what we can do for that? So they did a huge network meta-analysis from the available data, and they not only could be able to publish in the Journal of Laryngoscope, but when they returned to their hospitals, they immediately used it. And within a single year, 30% for the decrease. So there are plenty of knowledge from data generated in there, can be translated directly to patients. And we were also happy that Times Higher Education, which actually also putting together the universities list, they also found that this is an effective model. So pushing discoveries from the beginning with young, talented people and helping medical doctors and healthcare professionals to translate the data could be really helpful. So in this program, we have 5 distinct education elements: the group meetings, classes, project meeting, progress reports, which will occur next week when the students will present their data. But we are so happy, and this is one of the highlights of the program, the lectures by internationally distinguished scientists. And we are happy because this program has been established only like 5 years, but we had so many brilliant scientists, either Nobel laureates or like who are also had almost similar data and results in the field, and they are coming here to share their knowledge. And I have the privilege that this time we have 3 really, really distinguished speakers. So we will have Ole Petersen from the UK, who actually discovered so many things in the field of pancreatology, and our current knowledge in pancreatology wouldn't be there if he wouldn't put so many energy. We are also very happy to have Marty Chalfie here, who actually discovered the green fluorescence protein, which is actually now widely used in everywhere. And my special also welcome to Sharok Shariat, who in clinical science, I couldn't find any more inspirational scientist with unbelievable lot of data, and I'm sure you will enjoy it. And we always finish with a quote and with the students, and this is one of my best quotes, what Albert Einstein said, that if I am the smartest person in the room, then I am in the wrong room. Now, if I look at the young people's eyes with the huge amount of inspiration, if I look at our distinguished guests and what they are smart, I can convince them, and I'm sure that I am in a good room, because this is where I can learn from you and I can learn also from the students. Thank you very much for your attention.
Thank you, Peter, for the opening speech. And now I kindly ask our international guest, Professor Ole Petersen, to the stage to give his talk.
But before that, I will make an introduction about Ole Petersen, because I think it's good for everybody to know who is the distinguished guest. So, but before I start with Ole, we have to go back with one generation, because we are very proud that Ole half Hungarian actually, because his mother was Hungarian, and she was really a brilliant Elizabeth Klein, a pianist, a classical pianist in Budapest. And I'm also very happy that there are people also here who could enjoy the music she was playing. And actually she went to Denmark, to Copenhagen, actually in 1939, and this was the place where she and Ole Petersen's father actually met. And actually she was graduated in Budapest at the Franz Liszt Academy, and she received in 1966 the music prize of the Danish radio, and he in 76 from the Norwegian Composer Association. So she was really, really, really brilliant. And what the love brought together, actually, it's just completely changed the field in research in pancreatology. I remember my 90s when I started understanding the basic physiology of pancreatic acinar cells and ductal cells, and when I opened up the most distinguished publication, there was almost always one name in it, which was Ole Petersen, who actually was the very, very first time who understood how the cells respond with calcium signaling to a stimuli which is coming, and this is one of his most distinguished papers, Figure, which a model which actually described on how, like for acetylcholine stimuli, the cell will start via the calcium signaling, the exocytosis of the cells. So one of the biggest achievement for Ole is definitely the calcium signaling. However, the other one is, and I try to move forward with the pointer, but it's somehow not moving. Okay, so troubleshooting, guys, is one of the most important part of medicine. So we will have a look what we can do with it, because most of the experiments are fail, not only for the first time, but even for the second and the third one. So, and the next one, which is Ole really, really very famous from, is about the patch clamp techniques, which actually he was the very first one. He actually used the patch clamp technique in living exocrine cells. Thank you so much. And Yes. So he had really a major contribution to the understanding of the physiology of the ion channels, and we also had the privilege to welcome Erwin and Bert here also, and Bert Sackmann is actually the educational director of the Szeged program, which is here. They always said that this Nobel Prize would have go to 3 persons actually, and Ole was also mentioned among them. So there are, of course, plenty of titles and position what he had, and actually what amazed me when in pancreatology, that actually in 2010, around the age of I think 65 or something like that, actually when usually people retiring, he is definitely not retired. He even moved further in his career, and in the last 15 actually, or years, or 10 years, he is leading the Cardiff School of Biosciences and really achieved tremendous data. Concerning election of the national and international academies, if you look at the list, he's really not only a brilliant scientist, but a well-acknowledged one. I'm very happy to also mention that he is a member of the Hungarian Academy, actually, of Sciences, and he had plenty of selected awards, among them the gold medal by Academy of Europe, which is the highest recognition, and also the George Emil Palats medal, which is exactly 50 years ago was the discovery of the electrophysiology measurement of the exocytosis of the cell. So exactly 50 years ago, he received the Nobel Prize in 1974 in Stockholm. So we are also celebrating in pancreatology his Nobel Prize. But of course, and as I said, experiments usually fail not only for the first time, but for the second time as well. So it doesn't work again. So, but you know what is the difference between failure and success? That you tried one more time. So actually we will try one more time, and I am sure that finally we will reach a level when everything will move. So welcome in science. And I have only one slide left. Okay, it's working. Okay, now it works. Thank you so much. So if we come back one step, so Ole is one of the founding members of one of the most distinguished academies nowadays in Europe, which include over 5,000 members, and 92 of them actually received already the Nobel Prize, and many of them received after they became to be a member of the academy. And this recognition also for Ole was done by the Queen Elizabeth II for service actually to science. I have the privilege to work with Ole also, and we tried actually to go with the doctoral part what he has achieved on Essiner cells, and I think this kind of tango is working not only in cells but in friendship as well. So it was the very first time that we could invite Ole. He was here when we had the European Pancreatic Club meeting in 2009, and he continued to come back, and he was one of the key players of the 2012 establishment of the National Academy of Scientist Education. He's already visited the talented students, and even last year in the Semmelweis seminar, he was here. So he has a wisdom for young people. I don't know, you remember yours word or not? In a Bond film, he said that in which a police officer admits that he and his colleagues are perhaps not very clever, but they never give it up. So I think this is in science highly important not to give it up. So Ole, really thank you very much, not only for coming today, but your service in science, your service for Hungarian science, your service for Hungarian pancreatology. And thank you for spending your incredible, valuable time with us. And we are more than happy to listen to your presentation today. Thank you, Ole.
Well, thank you very much, Peter, for that over-generous introduction. But I appreciate very much, of course, the invitation to come here once more to one of the meetings that you always organize so well and which always are such a joy to participate in. So, what I hope to do today is to give some indication of what I would call a process of a discovery and also a little bit of process of application of a new knowledge. And if we move immediately to the next slide, I'm not sure which button to press, but here we are. So Peter, of course, has introduced this wonderful translational medicine program here at Budapest, which I've had the privilege of following for some years. And certainly there's no question that the application of knowledge we already have is insufficient at this point in time, and that Peter has always made that point very forcefully and effectively, how important it is to utilize all the knowledge we have. At the same time, we cannot be satisfied with the knowledge that we have. We need more. So one challenge which has sort of impressed itself on me for the last several years is that there is a big change if I look back at a perhaps already too long career. So when I was a young person starting out in science and very much in physiology as a subject, I think at the time I started teaching students who were only 1 or 2 years younger than myself, I actually had a reasonable grasp of the whole field of physiology. I remember every time a new issue of the Journal of Physiology came out, I would look at it, and I would not read every paper, certainly not, but I would know what was published every month, and I had a fairly good feeling for that now. I can't say that this is the case anymore. It's just not possible to follow all of that. And George Busacki in New York has made very eloquently the case that we are now in a situation where our personal ownership, so to speak, of the total knowledge that is available is very rapidly diminishing. So hundreds of years ago, people who were scientists and working in the field would actually know pretty well what was happening around it. Now we are personally only sharing a very tiny amount of the total knowledge that is available. So this has big consequences for the way we teach and the way we work. So our ability, as I put on this slide here, to extract and use relevant information is not only dependent on our ability to access the databases and so on, and we can learn about that from informatics specialists, but I think the ability to do that in a way that is meaningful depends actually on some general appreciation of the direction of travel of science. And this means that actually, contrary to what many people think, understanding the history of the progress of how science progresses is actually more important now, I think, than it was previously. Because only if you don't know the travel of direction, then you are just kind of having an instant picture of what things are now. You have no idea where is it coming from. You have no idea of which direction you are moving in. So I think that is important. It's also important to have a general feeling for the overall concepts, because otherwise you're just like a lay person who is searching in Google for some symptoms and will find that this is maybe this, maybe it is that. So that's the background for what I would like to do. So maybe the individual pieces of information I show you as examples are not that important for all of you, but I hope that the kind of process that I will describe will be at least serving as a kind of example of how things could happen. So the field that I will focus on today are ion channels. They are primary drug targets. Actually, a very large number of drugs actually are working by interfering with ion channels, either increasing the probability of channel opening or decreasing the probability of channel opening. And of course, ion channels are just one example of membrane transport proteins. So we basically have three different types: ion channels, pumps, ATPases that move things against electrochemical gradients, and various transporters that utilize gradients that are already existing. So when I started out in the 1960s as a medical student at the University of Copenhagen, in the transport field, the general notion at that time was very much focused on active transport. And this came, of course, from the laboratory of Hans Ussing in Copenhagen, which was totally separate from the Faculty of Medicine. So first time I met Hans Ussing was actually at a meeting in Britain, and I never met him in Copenhagen. I had a chance to that. And in my interest, for some reason, was in how transport is switched on and switched off. It was not a major preoccupation at that time. Most people who studied transport worked on intestine, on frog skins, on the urinary toad bladders, which are sort of transporting all the time, and there is some modulation of it. And for some reason, my interest was focused on the salivary glands, where you have this very sudden transition. And I was very impressed by this simple experiment that I started doing out. When you electrically stimulate the nerves to the salivary gland, the saliva is pouring out instantly almost when you watch it. You stop the electrical stimulation and secretion stops. And there was actually no knowledge about how does that happen. So I was thinking about this. So the only theory that existed at that point, and which is on the slide here, is that was a theory by a neuroscientist, actually, who had strayed into epithelial transport for a few years, Anders Lundberg, in Sweden. And he had the idea that what happened was actually that agonists, acetylcholine, adrenaline, whatever, would directly activate a chloride pump. And that fitted in with some of the work that happened in Ussing's group, where they published papers in the frog skin that adrenaline stimulation of transport would directly activate chloride pumps. So that was the overriding theory. And after publishing a number of quite unimportant papers, I finally did a meaningful experiment and looked at what the evidence actually was for this chloride pump hypothesis in the salivary glands. And so what Lundberg had discovered was that when you put a microelectrode into the salivary gland cell and you stimulate the nerves to the gland or stimulate directly with acetylcholine, the membrane hyperpolarizes, so the membrane potential increases. And he had some evidence that this might be due to increased uptake of chloride. So I did a very simple experiment. I replaced all the chloride with sulfate and expected this hyperpolarization to disappear. And instead of that, it actually became larger. So that did not seem to be immediately compatible with the chloride pump hypothesis. I also did an experiment looking at potassium transport. And as you can see in the lower right corner here, discovered that potassium is thrown out of the gland when you stimulate it. So I had the feeling that maybe what happened was not actually a direct activation of chloride pump, but actually opening of ion channels. Now, at that time, as I said, there were not many people thinking about this, so this did not have actually a very big impact. But some years later, I was for reasons which are still not very clear to me, I got this very honorable invitation to speak at a meeting of the Royal Society in London in 1970. And there I met for the first time another person who was thinking a bit along the same lines as myself, and this was Mike Berridge. Both of us actually gave our first talks ever to the Royal Society at that meeting, and it was the first time I met somebody who actually also, like me, was thinking that maybe activation of ion channels would play an important role. And Mike and I both worked on salivary glands, which was, of course, an extremely small field at that time. There was hardly anybody who was interested in it, actually. And our papers published after that meeting had very little impact. And while both of us had this intuitive feeling that ion channel control probably was important, none of us had actually a very clear hypothesis that we could put forward at that point. So over the years of this very long career, I mean, I don't know how many meetings I attended together with Mike Barrage, and I always felt, and I mentioned it at some point, that a meeting in this field, the importance of a meeting in that field was simply determined by whether Mike Barrage was present or not. If he was present, it was an important meeting. And sadly, of course, Mike died some years ago, and I was honored that I was asked to write his biographical memoir for the Royal Society. Now, a few years later, in 1973, I had the privilege of being able to organize a meeting at the Royal Danish Academy in Copenhagen, and I invited, of course, Mike to come there. And Mike had made fantastic progress in the 2 years. And for the first time, actually, he presented a coherent model for how fluid secretion in these insect salivary glands would actually work. And one key point was the activation of a chloride channel in the apical secretory membrane by an increase in the calcium concentration inside the cells. He also at that point had another chloride channel, the basal membrane, which he later wisely forgot about. And it was actually only very many years later that I was able to demonstrate very directly that this chloride channel exists very specifically in the apical membrane, when technology had developed in a sense and we could simultaneously measure the chloride current across an isolated pancreatic cell, and also at the same time uncage calcium in specific subregions of the cell. And we discovered that only when you uncage calcium near the apical membrane, you switch on the chloride current in the green trace, as you see on the right of the slide. So by using this technology of measuring calcium, of uncaging cage calcium in pre-selected locations of the gland, one could actually show very directly that this chloride channel exists there. It is specifically and exclusively in the apical membrane, and it is activated by a rise in the calcium concentration, and it is one critical element in the activation of fluid secretion, not only in the pancreas, in the salivary glands, in the lacrimal glands, in sweat glands, and also in the intestine. Now, meanwhile, of course, electrophysiology changed very dramatically, and Peter already alluded to this. In 1976, Erwin Neher and Bert Sackmann recorded the first tiny channels moving through a single protein channel in the membrane. And so this was obviously a breakthrough, because it was the first time that one could actually see the behavior of individual protein molecules in real time. However, as you can see on these recordings here, the recordings were rather noisy and not that beautiful, because the technique still was not fully developed. And for 4 years after this demonstration, basically nothing happened. Nobody really used the technique, including Erwin and Bert, because it was not very easy to use, and the recordings were by and large not very impressive. But in 1980, Erwin Neher made what to many people would appear to be a quite unimportant discovery, but actually turned out to be, from a practical point of view, enormously important. He discovered the gigaseal, which I've highlighted in red letters here. So he found that the seal between the glass micropipette and the surface membrane could be increased by orders of magnitude by applying a little bit of suction to the pipette. So we basically suck in a bit of membrane into the tip of the micropipette. And this seemingly relatively modest discovery was combined also with the realization that it only happened when the pipette was absolutely clean and had not been used before. Before that, everyone would use the pipette again and again, because that's what we did with sharp microelectrodes. You impale one cell, and you could use the same electrode for impaling another cell. But with the patch clamp type, it didn't work, actually. And so this combination of using a pipette only for the first time, applying a little of negative pressure, and Erwin personally discovered that by chance, but he was open in his mind and realizing this is actually a critical step. And it opened up the technique in a way that was totally unexpected, because not only was the electrical seal fantastic and gave rise to very clean recordings. And here you see one of the recordings by Bert Sackmann of the nicotinic acetylcholine receptor for the motor end plate. And you can just see how beautifully clean and clear these recordings are. So you can see there that the channel always opens up to exactly the same conductance state. The probability of opening can be predicted when you have gathered experience about the behavior of the channel, but it is a stochastic process, and nobody can predict that in 10 seconds the channel is going to be open. You can talk about probabilities, but we are looking at here the consequences of rapid conformational changes of the protein. Some of the conformations equate to an open state. Many of the other conformations equate to a closed state. So this was real progress. I happened to be in Göttingen to give a seminar that Erwin had invited me to give a few weeks after the publication of the Sigvars and Neer paper in Nature in October 1980. And I had thought that I was aware, of course, of the patch clamp technique for the Nature 76 paper, but I did not think that this was something that would be of immediate importance. Maybe I thought in the future I will think about it. But as soon as I saw the laboratory for the first time and saw what they could do with the technique, I immediately realized that actually the time is now, not in the future. So I went home immediately, wrote a grant application to the Medical Research Council because I needed different equipment from what we had at that time. And most importantly, the patch clamp amplifier was not commercially available at that time, so you had to construct it yourself. And so we got onto this fairly quickly. And as you can see in the lower right corner, we did actually the first, and Peter already alluded to that, the first recording of ion channels, single channel recordings in epithelial cells. And it settled for once this problem that many people had been discussing. Are there really ion channels in epithelia? Because for most people it was not very clear. In the nervous system, in the muscle system, it was quite clear. There are very, very rapid changes, and everyone knew well before the patch clamp technique, of course, that ion channels were crucial to the function of the brain and the nervous system. For epithelia, the general notion was that maybe ion channels have some importance in setting the membrane potential, but any acute regulation of ion channels was never thought about, actually. So we were able to show not only that the channels exist, but also that they work. And you have an interesting comparison here at the bottom of this slide. On the left side, you have the nicotinic acetylcholine receptor, which is activated directly by acetylcholine binding to the channel protein. And you need two acetylcholine molecules to bind to the acetylcholine receptor, and then it is in a state where it can open. In the pancreatic acinar cell, where acetylcholine also is a major stimulant, and as you can see, the recording configuration there was that we actually added acetylcholine outside the area from which we were recording, and then the channels opened. So it was quite clear that it was done in a quite different way. It was a messenger-mediated channel rather than a direct ligand-operated ion channel. A little later, we discovered the channel that actually turned out to be very important in epithelial cells, the calcium and voltage-activated potassium channel. And here you see the recordings. You can see that it's calcium sensitive, it is voltage sensitive, and you can generate this family of curves at different calcium concentrations in contact with the internal aspect of the plasma membrane. Under certain considerations, this channel is totally dominant in the pig pancreatic acinar cell, and this means that we could do quantification of the channels for the first time. We could record the whole cell recording, the WCR, where you see the total currents flowing across the plasma membrane. And they represent all the potassium channels that exist in the cell. So this total current is equal to the number of individual channels times the single channel current times the open state probability. So if the open state probability is 1, then the number of channels is the total current divided by the single channel current. And this way we were able to see the surprising discovery that actually the number of this particular ion channel in a single cell is very small, somewhere between 50 and 100 channels per cell. So this means that you have a chance when you put your patch clamp pipette on the cell surface to just isolate an area where there is only one channel. And you can then monitor exactly the movements of ions through this one single channel protein. So this enabled us then finally, combining all the things that I talked about, to have a coherent transport model. And I will not go in detail with this. The general takeaway message is that opening of chloride channels at the apical end of the cell and opening of potassium channels at the basolateral part of the cell allows the active transport to proceed so that fluid can be produced. And in the bottom of the slide, you see an old experiment of mine, which I actually carried out personally, on fluid secretion in the perfused rat pancreas. And you can see that when you stimulate the acinar cells, in this case with an analog of the hormone cholecystokinin, it's a frog peptide, carulin, it has exactly the same effect as acetylcholine, but acts on a separate receptor. You get this fluid and enzyme secretion, but the sustained phase of secretion is absolutely dependent on the presence of calcium outside the cell. Later, of course, we were able to record the actual calcium signals in the cell, and you see that to the left of the slide, where we actually record the calcium spikes from a human pancreatic acinar cell. And this is actually the normal physiological calcium signal, the repetitive short-lasting calcium spikes. Anyway, the fact that the sustained secretion is dependent on the presence of calcium outside the cell suggested that there is an important process of uptake of calcium into the cell, and that is important for the fluid secretion mechanism. So obviously one looks for analogies in cells that have been better investigated than a pancreatic cell. So here I give the example from the heart, and I've taken the recording from the left from a magnificent review written by Andras Varro, who I'm happy to see also here in the audience. And by the way, as a little digression, I would say if you want to understand how the heart functions, then you have to read this review in Physiological Reviews 21 by Andras Varro. And then you will understand how the heart functions, because this is a masterpiece of pedagogical writing. Actually, it's so clear and beautifully described and fantastically illustrated that I would say if you haven't read that one, then you probably don't really understand how the heart functions. Well, the point here is that the calcium entry in the heart is generated by voltage-gated calcium channels. You have the cardiac famous action potential described already in the 50s by Silvio Weidmann in Bern. And what you see is that the potential really changes dramatically in the heart, from in the example that Anders showed there, from minus 87 millivolts to plus 30 millivolts. So it's a huge shift of potential. And that activates the calcium current, and in the bottom left, you see the overall calcium current flowing into the cell. So that's sort of one model. You can also look at the single channel currents of these, and I've taken the example from another good friend of mine who unfortunately died some years ago, Harald Reuter. So could this work in the pancreatic cells? Well, actually not. Because in the pancreatic acinar cells, the depolarization is quite small, at most 20 millivolt or something like that. So even if voltage-gated calcium channels existed in the pancreatic acinar cells, it wouldn't work actually, because it's simply not a sufficient shift. As you see here in this recording, where we specifically focus on the calcium-activated chloride current, this is basically an electrophysiological recording of calcium spiking. And you see here that the beginning of the record, which was initiated by using the calcium-releasing messenger inositol trisphosphate discovered by Mike Barrett, which I mentioned before, it all occurs in the complete absence of external calcium. So the initial calcium signal is actually initiated by a release of calcium from internal stores. And there is not a dramatic depolarization. So, and the mechanism for this was, of course, clarified in '83, as I show in the inset in the lower right part of this diagram, where in experiments actually on permeabilized pancreatic acinar cells, Mike Barrett and collaborators, particularly Arjen Schultz, showed that IP3 releases calcium from internal stores. And a few years later, Andras Speth, who I'm again very delighted to see here in the audience today, characterized the receptor in the endoplasmic reticulum membrane that receives the molecule IP3, and by this interaction of IP3 with the IP3 receptor, the channel opens and calcium can be released from the internal store. So that's how it operates in the pancreatic acinar cell. Nevertheless, as I showed before, the sustained fluid secretion is actually dependent on calcium entry. So how does it happen? It does not happen by the method that I showed in Andras Varro's slide, because there are no voltage-gated calcium channels in these cells. So instead, that's a very complicated mechanism whereby calcium channels of a special nature are opened as a consequence of release of calcium from internal stores, as illustrated in this diagram. I shall not go through that in detail because there simply isn't the time to do that. But the point is that there is such a channel known as a calcium release activated calcium channel, a channel that opens up simply as a consequence of depletion of the intracellular calcium store. And finally, in 2013, we were able to measure this very, very tiny current. You cannot get single-channel recordings because the single-channel conductance is just too low for the patch clamp technique, but you can measure the overall currents that are done there. And moreover, and importantly, there are a number of small molecules that act as efficient inhibitors of this channel. Now, this has some consequence for a disease that both Peter and I have been much involved in talking about, because it turns out that whereas the physiological calcium signals, as I mentioned before, consist of short-lasting calcium spikes, if you generate a sustained elevation of calcium, as shown here in this slide here, then you actually destroy the cell. And it is what happens in the disease acute pancreatitis, where the pancreas digests itself. And basically there are two major causes of this disease. There is excessive alcohol consumption, or there are gallbladder complications. And here I just show the example of alcohol-related pancreatitis. We thought initially it would be directly caused by ethanol, but it is not. It is actually a combination of alcohol with long-chain fatty acids that generates fatty acid ethyl esters that causes a massive calcium release from the internal stores that then in turn activates the calcium entry through the mechanism that I just mentioned. And so we thought we should try and see, just as a proof of principle, whether inhibition of these crack channels could help against acute pancreatitis. And here you see a series of experiments we carried out shortly after having recorded these crack currents. And you can see that a particular small molecule, and there are a whole series of these small molecules that can do this, markedly reduce the calcium signal and makes it more physiological by short-lasting spikes rather than a major sustained elevation. It also inhibits very dramatically the intracellular protease activation, which is very dangerous, and most importantly, in a concentration-dependent manner, inhibits the extent of necrosis. So there's a proof of principle that this can actually work. And one of the reasons that it turns out is very effective is that it not only puts a brake on calcium entry into the Eisner cells, but also into other cells in the environment of the Eisner cells, stellate cells, which are illustrated there, which also have the same channels, and perhaps even more importantly, the macrophages that invade the pancreas in this initial inflammatory phase of the disease. They also have these crack channels. And by kind of reinforcing circuits between these three cells, we actually generate this very dramatic inflammatory process in the pancreas that actually will destroy it. In the very last few minutes of this talk, I will shift gears a little bit, but I will come back again to the main theme. And I'll shift now to cystic fibrosis, a disease that Peter also mentioned in his introduction, because it's also quite an interesting story. I talked very much about the calcium-activated chloride channel. Now we come to another chloride channel that also is very important. A key discovery was made very early and then totally forgotten about. And this was another very close friend of mine, is now long retired, Irene Schulz from Berlin. And she did experiments on her mentor, Karl Ulrich, who was the director of the Max Planck Institute Biophysics in Frankfurt. And she punctured the sweat glands of her mentor and were able to sample the primary secretion from the sweat gland, or the human sweat gland. And later she went to the NIH and did this study published in the JCI in '69, and discovered that in cystic fibrosis, the concentration of salt in the primary secretion was completely unchanged. It had been known for years, for centuries, that in cystic fibrosis, the sweat is very rich in salt. There is medieval poetry that speaks about the knowledge of mothers that when you kiss a baby, the baby tastes salty, it is a very bad sign, and that the baby will die soon. So medieval mothers just made that discovery by themselves, that a salty sweat is something really dangerous. And for many years, of course, that was the diagnostic test, simply a test of the salt concentration on the surface of the skin. So Irene tested this phenomenon, and it turned out that whereas the primary secretion was completely normal in cystic fibrosis, the final sweat was not. It had a very high salt concentration. And importantly, she also discovered that the electrical potential difference across the duct epithelium was increased in cystic fibrosis. So she reasoned that the problem must be that there is a low chloride permeability. So the sodium pump tries to absorb sodium chloride, but chloride cannot follow because the chloride channels are defective. So it was effectively the premature early discovery of a chloride channel defect in cystic fibrosis, but it had very little impact. Nobody paid attention to it. I think the paper is probably totally forgotten by even people who work in cystic fibrosis now. Of course, the cystic fibrosis field then really started to become important. The age of molecular biology and John Reardon's discovery in '89. that the defect is actually a very simple defect. There's a single amino acid missing at position 508. There are a lot of other mutations, but this is a principal one. However, that discovery did not itself indicate what the function was of the gene. So now you knew how it was composed, and that is the reason that it got this very, very strange name. So they named this protein, which is completely absurd, actually, cystic fibrosis conductance, transmembrane conductance regulator. And it's completely crazy because it's the normal protein has the name cystic fibrosis in it. And it is actually a normal protein. It has nothing to do with cystic fibrosis when it is normal. But the name stuck, and we are stuck with this name for what turned out to be a chloride channel. I became, in a very peripheral way, a little bit involved in it because one day, actually before this paper was published, John Reardon called me and said that he had kind of suspicion that this protein was a channel. And he wanted to send one of his collaborators to my laboratory to learn about a single channel recording. And this actually happened, and Christine Baer came to my laboratory in Liverpool at the time. She had never done electrophysiology before, but she had been kind of assigned by John Reardon that was her job to do that. So she stayed in my lab for about a year, and we published some papers on yet another cell type where calcium-activated potassium channels were there. It's not a particularly important discovery. And then she went back to Toronto. And then, a few years later, she, as I write there, created a sensation by actually showing for the first time that this protein is a chloride channel. So this paper in Cell, published in '92, is a massively cited paper, quite rightly. And perhaps it's one of these ironies that also is quite delightful in science, in a sense. In some ways, one could even say maybe this was my major contribution, that she came there and she learned a single channel, and she went back and she did something that actually turned out to be really important and had massive consequences for actually how the disease is treated to this very day. So what was very interesting was, she had this very nice recordings of this lipid bilayer where she incorporated this protein. And then there was another surprise. And then one assumed that in cystic fibrosis, this channel does not open. But this was not the case, because as you can see here in the recordings, even the delta F508 mutation, the channel does occasionally open, not as often as the normal one does. So actually it turned out that there are two defects. There is a defect in the processing and insertion of the protein in the membrane, because the cell's control system detects that the protein is not quite right and doesn't really want to insert it in the membrane. But there's also another defect, that even those molecules that do make it into the membrane actually don't open as often. And this meant again that there were two problems that had to be solved, and a massive number of small molecules were tested. And people found one type of agent that would improve the insertion of the faulty molecule in the membrane, and other molecules that would increase the probability of opening when the channel actually was there. And furthermore, so now there are actually three small molecules combined in one that actually, to a large extent, takes care of the problem. So it's a disease that now can be treated, and these patients that previously died, typically in their 30s, can now live a much longer and happier life. So in my final slide, I come back to the pancreas, because it turns out that this channel, CFTR, is a very important channel in the pancreatic ducts. And Peter, of course, has made his name originally by studying the processes in the duct. And you see here on the screen the transport model for the duct. I don't have time to go into details with it. But the key point is that there is in the apical membrane, also in the duct cell, like in the Eisner cell, a chloride channel, which is vital for the fluid production. But in the duct cells, the most important chloride channel is CFTR. And it is a cyclic AMP-activated ion channel. It's a completely different channel from the calcium-activated chloride channel. It has a different biophysical characteristics. It has a different selectivity, anion selectivity from the other channel. And it has also the advantage, it has a much bigger single-channel conductance. You can actually get single-channel recordings from the CFTR. And interestingly, it turns out that the duct cell is also not only important for the normal physiology in producing fluid secretion, it's also very important for acute pancreatitis because you need to wash out the enzymes that are activated inside the pancreas. And if you don't do that, then these enzymes become more active into the cell and they destroy the cell. And in a very beautiful paper in gastroenterology that Peter published in 2015, he demonstrated that these fatty acid ethyl esters will also have their effect in the duct cells. And more importantly and very interesting is that the alcohol has a very strong effect on this CFTR and it decreases the opening of that ion channel. And when we started working on the human pancreas, we discovered that in pancreas removed from patients who had obstruction of the pancreatic duct, the amount of enzyme activation in the Eisner cell was enormous. So this process of getting rid of the enzymes by washing them out is critically important for acute pancreatitis. And so this discovery again that Peter made, putting the importance, focus on the CFTR has very practical consequences. So I hope this gives you some impression of the development in a particular field. Here on this final slide to the left are the people who directly worked with me, and on the right side are the people who inspired me and continue to inspire me. Sadly enough, one of them, Mike Barrett, as I said, died some years ago. But both are very important, the people who actually do the work, but the inspiration you get from other people who work in adjacent fields is also enormously important. So I thank you for your attention.
Neither. All right. Okay, no, it works, I think. Thank you, Professor Petersen, for your really insightful presentation lecture today, really clearly highlighting the critical role of ion channels in health and disease. Thank you very much for this. I would like to open the floor for questions and a round of discussion. Any questions from our side or the other side? All right, not so far. I would like to start with one question. So once again, thank you very much for your presentation. What do you see, if you look at the horizon of your current research so far and the field of research, what will be maybe the next breakthrough in ion channel research?
Well, that's a very good question, of course. In some ways, technically, one could say very little has happened actually since the introduction of the patch clamp technique. There are a number of more automated devices now that can do it, but basically the technique of electrophysiology today is exactly as it was described by Avenir and Bert Sackmann in their famous 1981 methods paper. So technically I think it's very difficult. We are at the limit of resolution. As I already mentioned, there are some ion channels that unfortunately fall below the level of resolution. And at this moment of time, I don't think there is actually a physical possibility of increasing the resolution further. So from a technical point of view, there may not be such big insights. Of course, the big thing in ion channel field, which already started happening very soon after the patch clamp revolution, was of course the combination of molecular biology with electrophysiology. And the prime example of that was, of course, the fantastic collaboration between Bert Sackmann and Shosaku Numa at Kyoto University, where they did structural functional relationships and really characterized in a fantastic way the nicotinic acetylcholine receptor. So we have enormous amount of detail. We totally understand how that molecule moves and how all these things happen. And this, of course, can, so that is a kind of example from that spectacular collaboration. Unfortunately, Shosaku Numa died some years ago, of how the combination of molecular biology and electrophysiology can solve the most intricate problems of how the molecular configuration actually leads to conductance states or not. So that paradigm exists, and I think it is being applied, of course, to masses of other channels. And of course, we now have the three-dimensional structure of a number of ion channels that also gives. So in the future, there is obviously possibility for doing a lot of in silico studies, whereby you could predict what kind of molecules might interfere with ion channels. So I think there's a whole industry now in terms of looking at drugs that could interfere with various ion channels. The cardiac field is again one where, of course, there's an enormous amount of very important pharmacology, because there's a whole range of different ion channels. And I think in the future, as we get more and more information about the behavior of individual channels, you will be able to predict, actually, maybe even in some cases without initially doing any experimental work, but of course it would probably have to be confirmed in direct experimental studies, but you might be able to predict this molecule will actually be a likely candidate as an effective channel. So the number of ion channels is, of course, enormous. It's enormous superfamilies, and so there is an enormous amount of pathophysiology and studies there which will be applied. That's probably where the main thrust will be now for many years. So physiology has turned into pathophysiology, and we should expect a lot of real progress.
Thank you, Professor Petersen. I have one more question. Could you tell us more about what kind of challenges did you face during your research journey and applying the patch clamp technique back in the years, and how patient a young researcher should be in their research journey?
Well, I guess, as I think one person once said when he was asked about giving recommendations to young people, and he said there's only one recommendation, and that is to be lucky. Because luck is really, really important. And in my own case, I was, as such, incredibly lucky in a way that maybe most people would think that it was a stupid choice. I started working out with a very esoteric system, salivary glands that nobody was interested in. And most people would say, well, is that not a very clever way of starting out? But actually, it turned out to be very advantageous, because very early on, I became kind of seen as an expert in that area, because the competition was not very great. So that turned out to be quite lucky. With regard to the electrophysiology, I was just incredibly lucky that I got this invitation from Erwin Neher in the autumn of 1980. And the reason was that Erwin was becoming interested in the exocrine glands, actually, at the time. And they did actually quite a bit of work on the lacrimal gland. And he actually, being a very practical person, he just invited me because he wanted me to show them all the different exocrine glands, how to dissect out the different glands. And they realized that they didn't want to work on the same glands as I worked on, so they had the idea that they wanted to work on the lacrimal gland. But there was a problem in the rat and the mouse. The exocrine lacrimal gland is very close to the parotid gland, and some people actually made mistakes in published papers on the parotid that actually turned out to be the lacrimal gland. So Erwin was vaguely aware of that. So I said, why don't you come and give a seminar and you will do the dissection for us and show us where this different gland was. Was exactly what I did. But what is very important lesson here is that contrary to very many people who, when they discover a new technique, would like to keep it for themselves for a little while, to give themselves the opportunity to utilize the technique before competitors get going on. Erwin and Bert Sackmann did exactly the opposite. They were totally open, everyone who came to Göttingen. And when I came there to give my seminar, there were already quite a few other biophysicists who also were there. In fact, I shared a room with Bertil Hille at the time there because there were simply not enough guest rooms in the Max Planck Institute for everyone. And they showed everything. So Erwin and Bert gave me the drawings because, as I said, you could not buy a patch clamp amplifier. There was no commercially available one, so you had to construct it. But they gave me all the blueprints of how to do it. And by actually sharing this knowledge and by publishing very early on a very clear paper describing all the configuration of the patch clamp technique, which was published in a journal which is not very highly regarded, the Flüger's Archive, but actually is one of the most highly cited papers in biology because it just describes the patch clamp technique in such a way that people can actually apply it. But because I was there in 1980 and I got the blueprint and we actually could construct the amplifier straight away, we were able to do this. But it was sheer luck that I was there at that time, at the right place, at the right time. And from there on, it worked very well. And then I developed a relatively close relationship with Bert Sackmann, who was a very frequent guest in Liverpool and came to us again and again. And I was delighted, actually, later to introduce Bert Sackmann to Peter Hegge. And Bert then became involved in this whole education program. So from a single piece of luck, a lot of things flow from that, and that may also be a general lesson from all of that. You have to be lucky, but you also have to realize when you're lucky and understand that now is the time to move and to move fast, which is what happened.